Today, we're going to decode a biological language that is completely unlocking a new era of medicine. We are talking about peptides, the body's code, and how scientists are finally learning to write in that language to create incredibly powerful new drugs.
All right, so let's just dive right in with a question for you. When you think of the most advanced, most sophisticated machines on the entire planet, what pops into your head? You thinking supercomputers, maybe spacecraft, particle accelerators?
Well, the real answer is actually a lot closer to home. In fact, it's inside of you, right this second. The most complex, most elegant machinery we know of isn't made of metal and wires. It's biological, and it's running on a code written into our very own cells.
And at the very heart of all this biological machinery, you've got peptides. The best way to think of them is like the body's own private text messaging system. They're these short little chains of amino acids that carry critical signals, telling your body what to do and when to do it. And learning how to harness their power is sparking a revolution in medicine.
So, if these little molecules are so critical, how in the world do we make them? Well, the answer lies in a process that you can really only describe as a molecular assembly line.
The huge breakthrough came from this guy, Nobel laureate Bruce Merrifield. He invented a technique called solid-phase peptide synthesis, or SPPS for short. And as he put it, this invention totally changed the game. It turned a really complex, messy process into something streamlined and incredibly efficient. Honestly, the easiest way to picture SPSS is to think about building with Legos. It's that simple. You've got a solid base, and you just click on one brick, one amino acid at a time, in a super precise order, until you've built the exact thing you wanted. And this is what that assembly line looks like in the real world. I know, it might just look like some simple lab glassware, but inside that little vessel, scientists are meticulously building some of the most advanced molecules on the planet, one amino acid at a time. The whole thing boils down to this beautiful, elegant, four-step cycle. First, you attach the starting amino acid to a solid bead. That's like anchoring your first Lego to the base plate. Next, you uncap it, which basically just exposes a connection point. Then you connect the next amino acid, and then you just repeat over and over and over again until your peptide is totally complete. Now let's geek out for just a second on the chemistry. Don't worry about all the crazy symbols here. The key idea is this. Here's your solid support, and here's the first amino acid. This little P here is like a protective cap. That cap gets removed. That's the uncapping we just talked about. And that allows the next amino acid to connect. This is literally what's happening again and again inside that glass vessel. And why does this matter? Because this simple cycle is the key that unlocked our ability to create custom peptides on a huge scale, taking them from just a scientific curiosity to a real-world therapeutic possibility.
But, making a peptide is really only half the battle. How do you know for sure that you've made it correctly? This brings us to a huge challenge: quality control. And this is not a trivial step, not at all. As this quote from the research makes crystal clear, without really tough quality control, you can end up with data that is completely meaningless. It's literally the difference between a powerful drug and a useless powder. So this brings up a really important question. If you buy a peptide from two different companies and they both have the exact same sequence of amino acids, they should be identical, right? I mean, how different could they possibly be? Well, the answer is they can be dramatically different. Just look at this chart. This shows the immunoreactivity, basically how effective the peptide is, for the same exact sequence from three different vendors. And wow, this just says it all. Vendor A's product is more than twice as effective as vendor C's product. Same formula, wildly different results. The big takeaway here is that making a peptide drug isn't just about getting the sequence right. It's about manufacturing perfection. Quality matters a lot. Okay, so once we can build peptides and we can make sure they're high quality, the next logical step is to get creative. We can move beyond just copying what nature does and start designing super peptides. You see, the peptides that nature makes can be a little fragile. Your body is filled with enzymes that act like these tiny molecular scissors, and they're constantly breaking peptides down. That means their effects don't last very long. But by making some clever chemical tweaks, scientists can design peptides that are way more stable, more potent, and have much longer-lasting effects. And they have an entire toolkit to pull this off. For instance, they can link the two ends of the peptide together to form a circle. That's called cyclization,
which makes it much harder for those molecular scissors to chew it up. Or they can swap out parts of the chemical backbone for more robust structures. It's kind of like upgrading a wooden frame to a steel. So here's the really brilliant part. You don't have to modify the whole molecule. The key is for scientists to identify the hotspots, those few critical amino acids that are actually doing all the heavy lifting, the ones binding to a target. By focusing their efforts there, they can get the biggest bang for their buck. Let's look at a real-world example of this. This is a hotspot map for a protein interaction that's involved in autoimmune diseases. All these peaks and valleys show you how important each amino acid is for that connection. Now zoom in on this one specific residue, Y86. The data shows that a single mutation right there affects the molecule's adhesion by 1,000-fold. 1,000! That is a hotspot, and that's exactly where you focus your design efforts. The takeaway? Modern drug design is all about working smarter, not harder. By finding and tweaking these hotspots, we can turn fragile natural messengers into durable, potent medicine.
Which brings us to our final section, and really the ultimate goal of all of this, taking these brilliantly designed molecules from the lab bench and turning them into life-saving treatments for actual patients.
And today, this whole process is getting supercharged by massive computing power. Instead of endless trial and error in the lab, scientists can now design and test millions of peptide candidates virtually, which just dramatically speeds up the path to finding a viable drug.
This slide shows the difference perfectly. The top pipeline, that's the old way, screen a gigantic library of molecules and just hope you get a hit. It's slow, it's expensive, but the bottom pipeline is the new way, using computers to guide the design, starting with a much better pool of candidates and leading to a much higher success rate with way less effort. And the results are just undeniable. Right now, there are over 150 peptide-based drugs in clinical trials targeting everything from cancer to metabolic diseases. This isn't science fiction anymore. This is happening right now.
And many of these have already become billion-dollar blockbusters. Lupron, a peptide for treating prostate cancer, has been on the market for years. And you've probably heard of all the new GLP-1 agonists for diabetes and weight loss. Yep, those are peptides too. We're even starting to see the very first oral peptide drugs, which is a major milestone that people once thought was impossible. So we're left with this really incredible thought. Peptides are the fundamental code of biology. They're the messages nature uses to run the whole system. And for the first time in human history, we're not just learning how to read that code, we're learning how to write it ourselves. So the real question is, what problems are we going to solve next?